Recently, you may have heard about CRISPR, in the news or on social media. But what does CRISPR actually do? Does it toast things? Does it make British potato chips? What does this have to do with the designer babies we've been hearing about? Stick around and you'll learn the answer to these questions and more.
What is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Bacteria use CRISPR as an advanced defense mechanism against viruses, much like humans and other animals use specialized cells to attack pathogens. If one of the stored sequences of DNA is detected again from an outside source, then the bacterium can produce specialized proteins to find the new virus's DNA and cut it up into tiny fragments before the bacterium can be harmed. CRISPR consists of two components, a cellular scalpel that cuts DNA and the mRNA that serves as a guide to lead the scalpel onto a search through thousands of genes until it finds and attaches itself to the precise nucleotides that need to be cut out.
If this is only seen in bacteria, what is all the fuss about? While CRISPR has only been identified in bacteria,
Dr. Jennifer Doudna, a researcher at the University of California at Berkeley, discovered a way to manipulate the CRISPR system to alter the DNA of organisms other than bacteria. This discovery offered an unparalleled way to precisely change specific pieces of an organism's DNA. Before this discovery, gene editing would have cost months of work and thousands of dollars. Now it only costs approximately $75 and potentially a shorter amount of time with higher efficiency and effectiveness.
with this revolution in gene editing, it seemed that we could prevent many genetic diseases before birth and treat illness derived from damaged or faulty DNA in already born individuals, such as cancer. Unfortunately, the use of the technique on humans have not been fully approved yet. However, the technique is used in other fields. In agriculture, CRISPR is already in use, such as to edit genes in soybeans, rice, and potatoes to make them more nutritious and more resistant to drought. Can this be our new solution to world hunger? The technique can also potentially edit allergens out of foods like peanuts. Woo!
Although it is not allowed in the US, a research team in China managed to show that CRISPR could be used to manipulate the DNA in human embryos. However, this led to an increased scrutiny of gene editing technologies and raised many ethical questions that challenged the autonomy of the child, the non-maleficent aspect of practice, and the beneficence of the procedure. What would the test subject say about his or her genetic changes? How could this technology be used to improve individuals beyond the abilities of the average human? Could scientists theoretically engineer better athletes? Would the rich be able to use this technology to enhance their advantage over those who could not afford such a procedure? If you are interested in learning more about CRISPR, its history, its potential future, and more, please feel free to explore our website